So the eye is a G protein gated sensory receptor organ that we use to perceive light. The eye is going to be made up of 3 layers. From outermost to innermost, we have the sclera, the uvea, and then the retina. The retina is actually going to be made up of an inner layer and an outer layer, and we are going to show how that happens right here. So basically, you are going to have this optic vesicle that is protruding off of the developing diencephalon of the brain. So it's just going to be this evagination of diencephalon that is growing out towards where your eyes are positioned in the head. And as it's growing out, this front green part is going to kind of fold upon itself. So eventually you're going to end up with a cup inside of another cup that's going to form the 2 layers of the retina. The inner layer, the green layer shown here, is going to be the light-sensitive part of the retina. So that's going to be the layer that has the photoreceptors, the rods and cones that are going to sense the light hitting the back of the retina. This blue part, the outer layer of the retina, is the pigmented retinal epithelium, which does not have any of those photoreceptor elements. So the retina is all coming off of this diencephalon. So the embryonic connective tissue that we talked about here is the neuroectoderm. However, the uvea and the sclera are going to be formed from the surrounding mesenchyme that just comes and crowds around this retina and forms the surrounding uvea and sclera. Additionally, I want to point out that the protruding optic vesicle here, as it continues to form this optic cup, the part that is still attached to the brain back here is going to form the optic stalk that will eventually become the optic nerve. So the optic nerve is also a remnant of the diencephalon. So going back to the 3 layers of the eye, we talked about how the retina is going to be derived from the neuroectoderm, while the uvea and the sclera are going to be derived from mesenchyme, which is basically just a component of mesoderm in embryology that we haven't really covered yet. So when we're talking about the 3 layers of the eye, we know the sclera is going to be the outermost area of the eye, and we know that the retina is going to have the 2-layer cup that's going to be the innermost layer of the eye. But between them we have the uvea, and the uvea is going to be composed of 3 different parts. One is going to be the choroid. The choroid lies in the posterior 2/3 of the eye. It is highly vascular and pigmented, and the function of the choroid is to absorb light to reduce light scatter. That's why it's pigmented. Additionally, we have the ciliary body in the uvea. The ciliary body contains the ciliary muscle that is involved in controlling the thickness of the lens for accommodation. And then lastly, we have the iris. And the iris is going to be made up of 2 different muscles, the sphincter pupillae and the dilator pupillae, that are going to be involved in regulating the size of the pupil to regulate the amount of light that can come in through the pupil. So we all know that the iris is like what gives color to the eye, right? So let's take another look at the retina. So we already said how this green layer here is going to be the inner layer that's showing us the light-sensitive layer with the rods and the cones, and then this blue outer layer is the pigmented epithelium. So when we're talking about this light-sensitive layer in green, we are talking about the layer that the rods and the cones, or the photoreceptors, are located in. And they are actually located in the deepest layer of this light-sensitive retina. So the light has to travel through about 10 layers of cells before it can reach the rods and cones. You probably already know that the rods are going to function best in dim light, whereas the cones are going to function best when they're seeing colors or bright light. So basically, when you shine a light into the retina, what is going to happen is the light is going to travel through these 10 layers of cells before they reach the rods and the cones layer. And then it's going to excite the rods or the cones. The outer segment of the rods or the cones are going to function as a photon transducer, which is going to convey its input to the bipolar cells, which are adjacent to the rods and the cones, and then the bipolar cells are going to excite the ganglion cells. The ganglion cells are what are going to form the optic nerve by their axons collecting together and going out of the eye in the lamina cribosa. The lamina cribosa is just an opening in the sclera. In addition to the optic nerve, which is formed by the axons of the ganglion cells, the central retinal artery will also travel through this lamina cribosa, and that's a branch of the ophthalmic artery. So in most areas on the retina, there's going to be a convergence of rods and cones that are going to all converge onto a ganglion cell. So there may be one ganglion cell for 5 to 10 rods and cones. However, there's going to be one area in the retina where it's going to have a 1-to-1 ratio between cones and ganglion cells. And that area is going to be called the fovea centralis. So when you're looking at the back of the retina, the fovea centralis is going to be located lateral to the optic disc, and the optic disc is the area where the optic nerve exits the retina, and it's also known as the blind spot because there are no photoreceptor cells in that region. So if this is the nose, so that makes you realize that this is medial, whereas out here is lateral. The fovea centralis is going to be lateral to the optic disc. So the fovea centralis is going to be that area that has that 1-to-1 ratio of ganglion cells to cones. So it's going to be the area of highest visual acuity in the eye. Just surrounding the fovea centralis is going to be an area called the fovea, which is going to have some convergence, so it's going to have a little bit less acuity than the fovea centralis. And then surrounding the fovea is going to be the macula lutea, which is going to be a yellow pigmented area in the eye. So once again, keep in mind that all three of these structures are going to be lateral to the optic disc. Okay, so now we are going to talk about the visual pathway. And first, it's important to realize that you are going to have a right visual field, or things that you see to the right, and a left visual field, or things that you see to the left of you. And these are going to travel in various patterns through the brain to reach the primary visual cortex. The primary visual cortex is going to be located in the occipital lobe, and anything that you see in your right visual field is going to end up on your left primary visual cortex. Anything from your left visual field is going to end up on your right primary visual cortex. So that means you're going to have some decussations within these tracts. Additionally, your visual fields are going to be opposite of your retinal fields. So each of your retinas can be divided into 2 separate hemiretinas. You're going to have a nasal hemiretina that's located closer to the nose, and then a temporal hemiretina. So information that is coming from the right visual field is going to touch the nasal ipsilateral hemiretina and the contralateral temporal hemiretina. So as you can see, the right visual field is going to hit the left hemiretinas. And then the left visual field is going to hit the right hemiretinas. So it'll hit the left nasal hemiretina and then the left temporal hemiretina. So as you can see, the visual field and the retina field are going to be opposite. So anything from the left visual field is going to end up on the right retinal field. Anything from the right visual field will end up on the left retinal field. So from here you can see that the right visual field is already going to end up on the left side of the brain from the temporal side. However, it's going to end up on the right side of the brain when it comes to the nasal side. Therefore, this nasal fiber is going to have to decussate and cross over in the optic chiasm to end up on the left side of the brain. Additionally, you can look at the left visual field when it crosses over to the right eye's temporal retinal field. It's going to already be on the right side of the brain to go back to the right primary visual cortex. However, the left visual field that goes into the left eye on the nasal side is going to have to cross over through the optic chiasm to end up in the right primary visual cortex. So we can go ahead and draw that. These temporal fibers are not going to cross an optic chiasm, so they're going to come from the temporal retinas and just go out, whereas the fibers that are from the nasal side are going to have to come through and cross, as well as the fibers from the other eye. So anything that is seen before this optic chiasm is going to be considered the optic nerve. Anything after the optic chiasm is going to be the optic tract. So obviously you can see that the optic nerve is going to be composed of fibers from one eye from both the right and left visual field, whereas the optic tract is going to be composed of fibers from both eyes, but from the contralateral visual field. So anything after the optic chiasm is going to be from the contralateral visual field. And the ipsilateral retinal field, because this is the right optic tract coming from the right hemiretinas coming from the left visual field. The optic tract is going to synapse at the lateral geniculate nucleus, which is the visual relay nucleus of the thalamus. The LGN is going to be a 6-layer nucleus, and the first 2 layers are going to be the magnocellular layers, and layers 3 through 6 are going to be the parvocellular layers. The magnocellular layers are going to see contrast and movement, whereas the parvocellular layers are going to see color and form. So we know the LGN is going to receive fibers from both the ipsilateral and the contralateral eye. So layers 2, 3, and 5 are going to be ipsilateral from the ipsilateral eye. And then layers 1, 4, and 6 are going to be from the contralateral eye. So remember, both of these are going to be receiving input from the contralateral visual field. So if this is the left LGN, it's going to be receiving the right visual field and the left hemiretinas. So the medial portion of the LGN is going to receive fibers from the superior retinal quadrant, whereas the lateral LGN is going to receive the inferior retinal quadrant. So once again, as far as superior and inferior goes, their visual field is going to be opposite. So the superior retinal field is going to be the inferior visual field. The inferior retinal field is going to be the superior visual field. From the lateral geniculate nucleus, the fibers are going to project through the optic radiations to the occipital lobe. The medial fibers are going to end up being the most superior fibers in the optic radiations, and they're going to synapse in the superior bank of the calcarine sulcus. The fibers from the lateral LGN are going to be a little bit different. So the lateral LGN is going to have these inferior retinal fibers, and these fibers are going to travel through the optic radiations again to the calcarine sulcus. However, while they're traveling, they are going to loop anterolaterally over the temporal horn of each lateral ventricle. So this loop is going to be called Meyer's loop, and then they are going to end up at the inferior bank of the calcarine sulcus. So pay attention to the fact that these inferior retinal fibers going through Meyer's loop are going to have visual information from the contralateral visual field, and they are going to be the superior visual field because they're coming from the inferior portion of the retina. So moving back to the schematic, we're just going to show the LGN fibers that are from the superior retinal field going directly to it, whereas the inferior retinal field is going to have that loop, the Meyer's loop, whereas it'll be the same thing on the opposite side. Now remember, once you get back to the primary visual cortex, that's going to be Brodmann's area 17 and the calcarine fissure that's located between the cuneate gyrus and the lingual gyrus. So area 17 is going to be where the sensory information initially hits, and then areas 18 and 19 are in the cortical regions around the calcarine sulcus, and these areas are going to make sense of the visual information. So they'll analyze the color, the depth, the motion, etc. So one other thing to take note of is some of the fibers that are traveling with the optic tract will leave the optic tract to synapse at the superior colliculus. And those fibers are going to be the afferent limb of reflexive head and neck movements in response to light. Additionally, there will be fibers that leave the optic tract to synapse in the pretectal region, and those fibers are going to represent the afferent limb of the pupillary light reflex. So recall that the efferent limb of the pupillary light reflex would be the oculomotor nerve, which would constrict the pupil, whereas the efferent limb of the reflexive head and neck movements for the superior colliculus would be through the tectospinal tract. Okay, so now let's talk about lesions. If you were either stabbed in the eye or you had a lesion to the optic nerve, you would have the same visual deficit. So obviously the retinal field from the left eye would be obliterated, so you wouldn't have any information from the left eye. However, you would have both visual fields able to see in the right eye. Now, if you had a lesion at the optic chiasm, if it just went through the crossing fibers, you would get what's called bitemporal hemianopsia. So we know we're going to lose our retinal field from the nasal portion of both eyes, which is going to lose the visual field that's out in the periphery. So these medial retinal fibers see the most lateral portion of the visual field. So the lateral portion of the visual field would have a deficit. So sometimes you can think of this as like horse blinders in a race. Those horses so they can't see what's around them. And the buzzword for this particular scenario is when they're talking about the pituitary gland, because the pituitary gland lies next to this optic chiasm. So if you had a tumor of the pituitary gland, you could possibly have this visual deficit. Now, if you had a lesion in the optic tract, you would end up with a loss of the retinal field on the same side, because on this left optic tract, these are coming from both left hemiretinas, so we would lose the visual field on the right. So these are the left retinal fields that are taken out, which are both receiving information from the right visual field. So this is called a homonymous hemianopsia. So you would see this visual deficit if you had a lesion at the optic tract, at the LGN, or in the optic radiations before they're split or after when they are together. Lastly, if you had a lesion in Meyer's loop, so the question stem for this might say there's some sort of tumor in the lateral horn of in the temporal horn of the lateral ventricle, and that's because that's where Meyer's loop loops over. Remember, these fibers from Meyer's loop are carrying inferior retinal fibers. So we know these fibers are going to come from the inferior retina, and because we are on the left side of the cortex, we know that we are getting left retinal fibers. So if we are talking about a quadrantopsia, that is from the inferior retina on the left side, that means the visual deficit is going to be from the superior field on the right side. So a lot of times this visual deficit is called the pie in the sky because people just see this black spot in the top corner of their eye when they have a lesion in Myers-Leuk. Also, I illustrated these with the retinal fields shown in a circle and the visual fields shown in a square. However, it probably won't be like that on the exam. That was just so that they were differentiated. Another potential lesion could be a stroke to a primary visual cortex. And we know that if we are talking about a stroke in the primary visual cortex, we're going to be talking about the left retinal field and the right visual field. So if we had a stroke in the left primary visual cortex, it would look the same as the lesion in the optic tract. So that would be another homonymous hemianopsia, where the left retina is taken out and the right visual field is taken out.